Okay, so we are talking about chapter 2, and this is the what? Part 2 of chapter 2. And part 2 of chapter 2 talks about the biochemistry. So when we say biochemistry, we are specifically focusing on what? Specifically focusing on the our body, right? Our body composition. So our body, if you think about the chemical composition of our body, we have two different types of the compound, inorganic compound and the organic compound. So examples of inorganic compounds are probably your water, salt, acid, base, right? Okay. Can anybody tell me what is the difference between the inorganic compound and the organic compound? What about carbon dioxide, carbon monoxide? Okay, your book says that, but there are exceptions like carbon dioxide, carbon monoxide, which does not have the what? Carbon. But all the organic compounds actually do have carbon, carbohydrate, fats, protein, and nucleic acid. But what I understand, the difference between the organic and the inorganic compound is basically your inorganic compounds are very, very small molecule, right? Water, H2O, right? And if you think about salt like sodium chloride, NaCl, these are very small molecule. But when we say organic compound like carbohydrate, fat, protein, nucleic acid, they are like really large molecule. So let me actually show you an example, then you will understand. So when we say inorganic, this is your HCl. So see how small and simple it is. But if you look at one of the examples of what your organic, see, look at this, how large the molecules are, how large the molecules are. Look at this. Yeah, so your organic molecules are like really large. So look at this one molecule right here. So look at this molecule right here. So these are all organic molecules, which are very large molecules. Okay, so now let's start talking about the inorganic molecule first. If we want to talk about the inorganic molecule first, the first thing probably comes in our mind is which one? Water. Why? Because water is 60 to 80% of your body actually what? Made up of water. And then why water is actually such an important component or molecule in our body? Why? First of all, if you think about the properties of your water, they have a very high heat capacity. Does anybody have any idea what do we mean by the high heat capacity? Hmm? Um, you have to actually use a lot of temperature to change a little bit temperature change or the change of the state. Like for example, if you think about your body has 60 to 80% of the water. Now if you are walking outside, when like for example if you think about that, when the temperature turned into like negative, like maybe what, 20 degree Fahrenheit or something like that. So what happens? Do you think your water inside your body starts freezing? No, it does not. And whenever you are probably walking at the hot summer of Texas, like maybe 110 degree Fahrenheit, do you think your body starts boiling? No, it's not happening because of the high heat capacity of the water. You have to actually put a lot of temperature to be able to change the what state of the water molecule. So similar to that, high heat of vaporization. Have you ever saw boiling water? So to be able to boil the water, you have to actually put at least 100 degrees Celsius. That's a lot of temperature to actually change the state of the what? Water molecule. So everyone understand about the polar solvent properties, right? Yes or no? We just discussed about it. So when we say polar solvent properties, what do we mean by that? They are not equally sharing the electron. Since they are not equally sharing the electron, they have the what? Partial positive and the negative charge. Due to that partial positive and negative charge, they're very, very what? Welcoming, right? Whoever comes in the reaction, they would actually hug them, welcome them very easily. Okay, and everyone loves water because water loves everyone except hydrophobic, of course. Hydrophobic is scared of water. Okay, and then reactivity. So one thing I want you to understand about the reactivity, this is very important. I don't know, we will talk about it later, but for now I want you to understand a lot of reaction in our body is actually dependent on water. A lot of the time we release water during the reaction. A lot of the time also we use water to what? Go through the reaction. So what does that mean? A lot of the reactions are actually dependent on the water. That is called the reactivity. And when we say cushioning, what do we mean by that? So protecting, like for example, if you think about your brain is in the what? Cerebrospinal fluid, right? Which is protecting and cushioning. If you think about your all the cavities inside your body, they all have the what? Serous membrane, double membrane. And between the serous membrane, you have the what? Serous fluid. Remember? Okay. So this serous fluid has a lot of water. What is it providing? A lot of cushioning to the organ. Okay, so now you understand why water is such a useful molecule in our body. So our God or creator was so like intelligently actually made our body, right? So that like it it is like really functional and effective. Okay, so now let's talk about the salt. So when we want to talk about the salt, you need to think about every time you make the ionic compound, whatever compound you have made on the paper today, all of them are actually what? Salt. Okay, so ionic compounds are always salt. So one of the important understanding of the ionic compound we just talked about, every time you have the ionic compound or salt, they are very easily what? Get dissociated in the water molecule. That is the reason ionic compound is what? A little bit less stronger than the polar non-polar covalent bond. Okay, so as you can see, very easily your ionic compound gets dissociated in the water and turns into what? Positive ion and the negative ion. Okay, so the next one we are going to talk about acid and base. Actually, I want to add something about the salt as well. A lot of the salt in our body, do you know salt is very important for our body? Do you know why? Because they get dissociated into what? Ions. When they are getting dissociated into ion, we call them electrolyte because they can conduct the electrical current in the solution. So that is the reason salt is very important part of your diet. You have to have some amount of salt in your body to be able to provide the what? Electrolyte homeostasis in your body, so that your body can actually function efficiently. Okay, yeah. So now let's talk about the acid and bases. Have you ever heard of the term acid and bases before? Does anybody have any idea what is acid? pH lower than 7. Anything else you want to tell me based on the information you have learned so far, not the new information? Positive charge, Anything else? Nobody wants to tell me anything? So when we say acid, acid always have the what? Hydrogen in the molecule. That doesn't mean that if they have hydrogen, they are acid. When we call them acid, we call them acid whenever they actually what? Donate the hydrogen ion. They donate the what? Hydrogen ion in the solution. Whenever they donate the hydrogen ion in the solution, we call it what? Acid. Okay, for example, hydrochloric acid, HCl, in the reaction or in the solution, they're going to give out hydrogen ion. One thing I want you to understand, when we say hydrogen ion, what do we mean by that? Usually in normal situation, hydrogen has how many electrons? One. If hydrogen, when we say hydrogen ion, how many electrons do they have? When we say hydrogen ion, huh? Zero, because hydrogen ion has one electron, right? They will give it out. If they give it out, they do not have any other electron left. And hydrogen does not even have any neutron. So what does that mean? They only have one proton. That is why hydrogen ion is also called proton. So when we define acid, what is the definition of acid? Acid is basically the what? Proton donor or hydrogen ion donor in the solution. Does that make sense to everyone? So what is the definition of acid? Proton donor or hydrogen ion donor. So now what about the bases? Bases are actually, they're gonna accept, they're gonna what? Accept the proton or hydrogen ion, what? Provided by the who? Acid in the solution, right? Sometimes they can also give out hydroxyl ion in the solution, but not all the time. Okay, some bases can actually also give out hydroxyl ion in the solution. For example, sodium hydroxide is a base which can actually give out what? Hydroxyl ion, but not always. A lot of the time, for example, if you think about the bicarbonate, ammonia, so these are all, they do not have the hydroxyl ion to give out, but they can still pick up the what? Hydrogen ion from the solution. So that is why they are called the bases. Now let's talk about the pH. She was saying that when we say acid, acid has the low pH. So before I talk about low pH, let's talk about what is pH. When we say pH, what do we mean by that? pH is basically a scale which measure the concentration of the hydrogen ion in the solution. Okay, the more hydrogen ion you have in the solution, more acidic it is. So whenever in the solution you have a lot of hydrogen ion, you call it pH 0. pH can be 0 to 14. pH can be what? 0 to 14, and 0 to 6.9 is actually indicating the acidic pH, 7 is neutral pH, and 7.1 to 14 is indicating the what? Basic pH. Okay, clear? Okay, so one more time, 7 is what? Neutral. 7.4 is slightly basic. So the reason I am saying 7.4 is a very important one, because our blood pH is what? 7.4. And maintaining that specific pH in our body is very, very important. Even slight change of the pH, like for example, from 7.4, if it turns into 7.8, you can actually die. So that important it is. So what does that mean? Now I have to talk about another word which is called buffer. What is it called? buffer. So when we say buffer, what do we mean by that? It means that these are the molecule which will prevent the change of pH. Which will prevent the what? Change of pH. So buffer is a molecule which prevent the change of pH. So how does they do it? Like for example, one of the way to do it, like for example, your pH rise. If your pH rise, what does that mean? You have to add hydrogen ion, right? When you add hydrogen ion, pH decreases, right or no? The more hydrogen ion you have, the less pH you have. So if you have higher pH, that means you have less hydrogen ion in the solution. So you have to increase the what? Hydrogen ion. So for example, this H2CO3 is a hydrogen ion donor, will give out the hydrogen ion so that it increases the what? pH, right? Now, for some reason, you have what? Low pH. Now you want to increase the pH. If you want to increase the pH, what you have to do? You have to decrease the hydrogen ion concentration. To decrease the hydrogen ion concentration, you need an what? Hydrogen ion acceptor, which will accept the hydrogen ion so that your what? pH comes to the normal, right? Okay, so that is the function of buffer. So one more time, what is buffer? Buffer is a molecule which prevent the change of pH. Okay, clear? Okay. So now let's talk about a reaction which is called the neutralization reaction. So when we know that acid has what? Acidic pH less than 7 and base has pH more than 7. So now when we say neutralization reaction, what do we want to do? We want to make something which has neutral pH, which is 7. Usually salts are what? Salts have the neutral pH. So when you add a base and an acid, you can actually make the what? Salt and water. This salt and water both have the what? Neutral pH. That is why this is called neutralization reaction. Okay, so now let's see how much you can answer. Which property of water contributes to body temperature regulation through sweating? Which property of water? So we are asking about the property of water. So we have learned about so many properties of water, right? Huh? High heat capacity or high heat of evaporation? High heat of evaporation, because how the sweating works. So when you have high temperature in your body, you sweat so that the sweat get evaporated. When the sweat get evaporated, it takes a lot of heat from your body, right? That is how your body can actually what? Can cool down, right? However, during the humid day, do you think this is very efficient? Not very efficient, because it is so hard to evaporate. If you cannot evaporate, you cannot actually reduce the temperature of your body. So what type of substance release hydrogen ion when added to the water? Acid. Very good. So what is the general relationship of hydrogen ion and pH? The more hydrogen ion you have, less pH you have. Very good. What are buffers important? Why are buffers important? It resists the pH change. What is the pH of our body, of our blood? 7.4, slightly basic. Okay, and maintaining that specific pH is very, very important. So now let's move forward and talk about organic compound. Do you guys know what are the organic compounds in our body? What are the organic compounds in our body? Carbohydrate, lipid, protein, nucleic acid. Very good. So these are the very important organic molecules in our body. So when we talk about these organic molecules, I want you to understand one very important thing, which is all these organic molecules are very, very large. They are very, very what? Large. If they are very, very large, what does that mean? It means a lot of them are actually what? Polymer. A lot of them are actually what? Polymer. So if we say polymer, what does that mean? They also have monomer. So they also have the what? Monomer. So exactly like if you think about one of your one of your organic molecule is like the wall, the brick by brick is making the wall. What does that mean? The wall is the polymer and the bricks are the what? Bricks are the monomer. Clear? So out of the 4 organic molecules in our body, 3 molecules in our body are true polymer. Which are those 3? Carbohydrate, protein and nucleic acid are the what? True polymer. That means they have the what? Monomer. You can actually compare them with the wall and they have the brick, like polymer and the monomer. However, lipid is a very large molecule. However, they do not have the what? They are not true polymer, because you can think about it like this. If you think this wall is the what? Polymer and they have the monomer, what about this ceiling right here? In this ceiling, we not only have this, we also have the what? The light. So that means we cannot really say they have the what? Monomer, because even though this is very, very large, but they have so many other things which are making up this, right? They not only have the bricks but also other things. A lot of other things together is making a large molecule. Does it make sense? So do you understand the difference between the polymer, monomer, and when we say lipid is not the what? Lipid is not the what? Polymer. Lipid is not a lipid is a large molecule, but this is not a what? Polymer. So one thing I want you to understand, when we say monomer, what does that mean? The same type of molecules, you are putting all these same type of molecules together to make the what? Polymer, right? So for example, same molecules, 3 of the same molecules, I am making a bigger molecule right here, right? So you are basically, from monomer, you are synthesizing polymer. So what you are doing? Synthesis. What you are doing? Synthesis. During the synthesis, you have to always release a water molecule. You have to always release a what? Water molecule. That is why when you make polymer from monomer, this process is called dehydration synthesis. What do you call it? Dehydration synthesis. So what is dehydration synthesis? You are synthesizing polymer from monomer by releasing what? Water. Okay. Now let's talk about when you want to break down polymer. You have a polymer. Now you want to what? Break it down to what? Monomer, right? That means you are breaking it down, which is what? Lysis, right? So lysis. During the lysis, during the breakdown, you actually have to add water. That is why this reaction is called what? Hydrolysis. That is why this is called what? Hydrolysis. And when you break down bigger molecule to smaller molecule, you call it hydrolysis. When you synthesize larger molecule from smaller molecule, you call it dehydration synthesis. Clear? Yeah? Okay, good. So now let's start talking about the process I have just showed you, right? For example, you have one monomer, another monomer. What is happening? You want to add them together. You have to release the water molecule from there to make the bond. Okay? So that is what is called the dehydration synthesis. And when we say hydrolysis, you want to break down this bigger molecule, you have to add water so that you can separate them. Okay. Okay. So now let's start with the first organic compound. We call it carbohydrate. First organic compound, we call it what? Carbohydrate, which is a very important molecule in our body. So when we say carbohydrate, so first of all, let me tell you something very important. We know that we have our carbohydrate and then we have polymer. Like remember, you ate the bagel, right? In the morning, you ate the bagel in the morning, which had the what? Starch. So starch is a what? Carbohydrate, and it is an example of what? Polymer. It is an example of what? Polymer. So what is the monomer? Glucose is the what? Monomer. So by the digestion, what you are doing? You are breaking the starch down to what? Monomer, which is the glucose. Then the glucose is going to get into your cell and then you are gonna generate the what? Energy, right? Okay. So now what I want you to understand right here, you can have this polymer which is starch because she was eating a plant carbohydrate, that is why that was starch, right? Now what if she drank milk together? Milk also has the what? Carbohydrate, we call it lactose, right? So when we say starch, starch has a lots of glucose. That is why we call it polysaccharide. What do we call it? Polysaccharide. But when she ate milk with it, milk has lactose, and lactose is basically what? A disaccharide. When we say disaccharide, what do we mean by that? It has two monomer, not one, what? Two monomer together, not multiple monomer together. Does it make sense? So your starch is made up of so many what? Glucose. And your lactose is made up of what? Only two monomer. One is glucose, another one is galactose. So everyone knows we take a lot of sugar, table sugar. Table sugar is also what? Disaccharide. It has two monomer. Those monomers are glucose and fructose. Okay. And then another example could be, we talked about the lactose, sucrose. Sucrose is in the table sugar. And then what else? Glucose and fructose together make the sucrose, and glucose and galactose together make the lactose. Can anybody tell me another disaccharide? Glucose and glucose together make the maltose. Okay, so these are called the disaccharides. So when we say disaccharide, you are adding two monosaccharide together. And when we say polysaccharide, what does that mean? We are adding multiple monosaccharide together, more than two. Okay, so in the plant, they produce what? Starch. And in human body, for example, instead of eating one bagel, what if I ate one bagel, 1 cup of orange juice, and then probably 2 sandwich? What will happen? I have a lots of glucose, and you do not need that much of glucose for your energy. What are you going to do with it? Anything else? Okay, so now let's talk about a little bit more important understanding right here. So when you eat glucose, the first priority is to use the glucose to generate the what? Energy. Now whenever you are done with the energy, what are you going to do? If you have, for example, once upon a time I used to make like maybe $2,000, and with the $2,000 I'm done with all the money because I have to pay for rent, car, everything, and I'm done. I have no money left, right? Like for example, now I can make like $4,000. Now I have $2,000 extra. What do? I would probably put them in the saving account, right? So in your body, you also have the what? Saving account. That saving account is what? Your liver and your muscle. So you actually put all those extra glucose together in your saving account as a polysaccharide called glycogen. Have you ever heard of that? Glycogen. Glycogen is the what? A polysaccharide which you store in your liver and muscle for as a saving. Okay, but that saving is what? Very easily accessible. What does that mean? Whenever you need it, you just move it from your saving account to what? Checking account and use it, right? Very easy. But what if you have even more? You probably want to put them in the Roth IRA, or maybe you want to put them in as a fixed deposit, right? That is your fact. So where that's not very easily accessible, you have to put them for the what? Long-term storage. But the liver and the muscle you have, where you can actually what? Put your glycogen for easily accessible storage. Like for example, I did not eat anything from the morning yet. How I am surviving? The reason I can survive because my liver and muscle had some glycogen storage from yesterday. So that is what I'm utilizing right now. Does it make sense? So what is the storage molecule inside our body and storage polysaccharide inside our body? Glycogen is made up of a lot of glucose. Clear, everyone? Yeah. And glycogen is an example of what? Polysaccharide. And it's made up of monosaccharide. What is the name of the monosaccharide? Glucose. The name of the monosaccharide is what? Glucose. Okay. So now we have learned about the monosaccharide, we have learned about the disaccharide, we have also learned about the what? Polysaccharide. So how you put all the glucose together and make a really large molecule in your liver and the muscle to make the what? Glycogen. So let me tell you also one more interesting thing which is not important for you. Your liver is very helpful. Whenever you need the glucose, they're gonna give all the glucose to all over your body. But you know what? Your muscle, they're very, very selfish. Do you know why? Because they do not share their glycogen storage with anybody else. They just keep it for themselves. They want to use it for themselves because your skeletal muscle is very, very what? Very highly energy consuming part of your body. So they do not want to share that with anybody. They keep it for themselves. I thought it was interesting. Just that's why I told you. Okay. So uh, now let's talk about next organic compound. I would say this is the most important organic compound in your body because this is the compound which is the basic structural molecule in your body. Because all the collagen you have, all the hemoglobin you have, all the enzymes you have for the function, everything, all the proteins in your body, right? This is the proteins which is making your body up, right? Which is a very important basic structural molecule. What is the name of the basic structural molecule of our body? Protein. Okay. So now let's talk about the protein. So protein is also an example of what? Polymer or not? Protein is an example of what? Polymer. Like for example, if you think about the collagen, collagen is an example of what? Polymer. So what is the name of the monomer? You should be able to tell me because we did talk about it already before. What is the monomer of protein? Amino acid. What is the monomer of protein? Amino acid is the monomer of what? Amino acid is the what? Monomer of protein. Okay, amino acid is the monomer of protein. So do you know how many amino acid we have in our body? 20 amino acid. In our body, we have only 20 amino acids. So based on the instruction from your DNA, you put them together to make different proteins in your body for you to give you what? To have a structure in your body to function also. All the enzymes, all the hormones, most of them are what? Made up of the protein. Okay. So now protein contain both an amine group and an acid group, can act as either acid or base and differ by which of the 20 different R groups it's present. Based on that, they will have their what? They will have their specific criteria. So now we have already learned that two monomers together bind with each other to make the what? Polymer. So when they make the polymer, what do they do? Synthesis. So they have to release the water, dehydration, synthesis. And the name of the bond they have in the amino acid, in the protein, the name of the bond is called peptide bond. Peptide bond. That is why a lot of the time when you are putting a lot of amino acid together, that is also called what? Polypeptide. Okay. The reason this is called polypeptide, because the amino acids are actually what? Bound to each other by a bond called peptide bond. So now when you think about the protein has four different types of the structure. Protein has how many different types of the structure? Four different types of the structure. So the first of all, do you remember when you have the instruction in the DNA? So what do? You make the messenger RNA, and then your messenger RNA, based on the messenger RNA information, your transfer RNA go and bring all the amino acid and put them together and make a polypeptide chain. That polypeptide chain is called the what? Primary structure of the protein. The polypeptide chain is called what? Primary structure of the protein. What about the secondary structure? When that primary structure can make a chain or a sheet, we call it alpha helix or beta sheet. These are called the what? Secondary structure. Now let's talk about the tertiary structure. So as I told you, your protein can have positive and the negative charge based on the whatever amino acid they have. So now some part of it have positive charge, some part of it has it negative charge. So due to the charge in the different parts of the polypeptide chain, they will fold. Okay, they will do the what? Foldings. They will have the foldings. When they fold, we call it what? Tertiary structure. And what about the quaternary structure? In the quaternary structure, more than one polypeptide actually what? Fold with each other to make the what? A quaternary structure. So now let's talk about something very important, which is called denaturation. Like for example, if you have eaten an egg in the morning today, anybody? No, okay. Whenever you eat egg, what do? You cook it, right? Or you just eat it raw? You cook it. So when you cook it, you are actually denaturing the what? Protein in the egg, right? Now, my question for you, if you degrade or denature the protein in your egg, what is the purpose of eating that egg? For example, she ate bagel so that she can get some glucose, right? You eat egg, meat so that you can get some protein. Now, you ate egg and you cooked it and you denatured the protein. What is the purpose of eating that egg? It still has some protein. I am looking for a different answer. Hmm? Huh? What is the monomer of protein? Amino acid. Yeah. You do not eat protein for getting protein. You eat protein for getting amino acid. Your body doesn't care which protein you are eating. Your body only cares if your body has enough amino acid to make the protein based on the instruction you have in your DNA. Does it make sense? So for example, now you want to make your hair better, skin better, you are eating collagen. Do you think the collagen is going to be collagen in your body after you eat that? No. It will first get broken down to amino acid. If you have instruction in your body to make the collagen, that is when they're going to use those amino acid to make the what? Collagen protein. Does it make sense? Yeah? Okay, good. So what I'm trying to tell you, you can very easily denature the protein by what? Heating, changing the temperature or changing the pH. Okay. Okay. So now let's come and talk about the nucleic acid. So when we say nucleic acid, what do we mean by that? All the DNA and RNA. DNA and RNA. So when we say DNA, what do we mean by that? Deoxyribonucleic acid. And then when we say what? RNA, what do we mean by that? Ribonucleic acid, right? So what is the difference between the DNA and RNA? We will talk about it, but before that, I want to make sure that you understand your nucleic acid is what? A polymer. So what does that mean? It has a monomer. What is the name of the monomer? Nucleotide. Nucleotide is the monomer of who? Nucleic acid. So now if you have nucleotide as a monomer, so now let's look at the structure of the nucleotide. When we say nucleotide, in the nucleotide you have a pentose sugar, you have a phosphate and you have a base. That base, remember in the DNA you have adenine, guanine, cytosine, thymine, and in the RNA you have adenine, guanine, cytosine, uracil. And what about the pentose sugar? This pentose sugar, in your DNA you have the deoxyribose sugar and in the RNA you have the ribose sugar. Okay. And when we look at the structure of the polymer, in the polymer they are going to make one strand of the DNA, two strand of the DNA, and both of them are what? Anti-parallel and complementary to each other. Who remembers what does that mean? We did talk about it before. What does it mean by complementary and what does it mean by anti-parallel? Yeah, one is 5 prime to 3 prime, another one is what? 3 prime to 5 prime. But do you guys understand what does that actually mean? So if you think about the one strand of the DNA, they have this sugar, right? They have this sugar, and this is the pentose sugar. That means it has 1, 2, 3, 4, 5 carbon. How many carbon? 5 carbon. So in the 5 carbon, carbon number 5, they have the what? Phosphate group, and this carbon number 3, they have the what? Base, right? So now if you make it like this, I can just draw it maybe like this. So what do we see? The 5 prime on the top and 3 prime on the bottom. So this is called the what? 5 prime to 3 prime. And when we say what? 3 prime to 5 prime, one is 5 prime to 3 prime, another one is the what? In the opposite direction. So this one has the phosphate on the bottom, 5 prime on the bottom, and then this one has the what? Maybe T, right? Doesn't make sense? So now this line has the phosphate on the top, this line has the phosphate on the bottom. So this is 5 prime to 3 prime, this is 3 prime to 5 prime. That is why this is called the anti-parallel, and the reason they are called the what? Complementary, because in one strand if they have T, another strand they have the what? A. If this one has T, this one has A, this one has A, this one has T. So that is why they are called the what? Anti-parallel and complementary. ATGC. A binds with T and G with the C. Clear? Is it clear now? Okay. So now we know about the DNA. What is the difference with the RNA? RNA is not two-stranded. RNA is what? How many stranded? One strand. And what is the name of the sugar? Ribose sugar. And they have all the three different bases except the thymine. Instead of thymine, they have the what? Uracil. Okay. So now let's talk about another nucleic acid in our body, which is a very important one, which is called the high energy molecule, right? We all have heard of what? ATP, energy currency in our body. To be able to survive in this world, you need money. To be able to do all the function in your body, you need the what? ATP, adenosine triphosphate. So if you look at the adenosine triphosphate, you see that they also have a pentyl sugar, they also have adenine base, and they have three phosphate. This phosphate is actually very high energy molecule. Whenever the more phosphate you have, the more energy you have. So every time you break this phosphate bond, what do? You release a lot of energy. That is the energy you actually use for function. If they have 3 phosphate, we call it adenosine triphosphate. If they have 2 phosphate, we call it adenosine diphosphate. If they have 1 phosphate, we call it adenosine monophosphate. So for all the function, do you remember when we talked about the membrane transportation? Your sodium potassium pump requires what? Energy, ATP. For me to walk, contract and relax my muscle, I need the what? ATP. For chemical reaction to happen in your body, you need the ATP. Okay. Okay. So now let's talk about a very important organic compound in our body, which is called lipid. So lipid, what is the difference with the lipid with other organic molecule? Lipid is not polymer. Lipid is not what? Polymer. Instead, lipid is a very large molecule. It can have carbon, hydrogen, oxygen, phosphorus, but they are scared of water. They are insoluble in water. But there are many types of the what? Lipid in our body, right? There are many types of the lipid in our body. For example, triglyceride, phospholipid. We have learned about that in the cell membrane, right? And then we have the steroid, all the cholesterol you have heard of, right? Your cholesterol is what? Steroid, vitamin D, all this. And then icosanoid, these are the what? This can make the prostaglandin. A lot of the function is actually dependent on the prostaglandin, right? We are going to talk about those. So now let's talk about the triglyceride first. So when we say triglyceride, it has the what? 3 glycerol and then 3 fatty acid and a glycerol molecule make the what? Triglyceride. So this triglyceride is the triglyceride we actually use in our cooking, right? Have you ever used oil to cook? Yeah. Have you ever used butter to cook? Yeah. So now this butter and oil are the example of the what? Triglyceride. So if at room temperature your triglyceride is, if it is what? Solid, that means that is saturated fatty acid. If it is liquid, that means this is unsaturated fatty acid. So when we say saturated fatty acid, what do we mean by that? It means all the hands of the carbon is actually fulfilled by the saturated by the what? Hydrogen. Okay. So that is why they are called the saturated fatty acid. And what about the unsaturated fatty acid? They have the what? Double bond. They have the what? Double bond. When they have the double bond between the carbon, what does that mean? Some of them, their hand still is not actually what? Holding the hand of the hydrogen. So they still have the capability to hold more hands of the hydrogen. So that is why they are called the unsaturated fatty acid. So the unsaturated fatty acids are all the oils you get, like the plant oils. One of the heart healthy one is the what? Olive oil, because it has the what? Monounsaturated fatty acid. So there are also what? Trans fat. There are some unhealthy trans fat. When we say trans fat, what do we mean by that? It means that you are actually converting your unsaturated fat to saturated fat by maybe heating again and again, or hydrogenating and all this. So all the oil used in what, maybe McDonald's or like over and over, you are basically making the what? Trans fat out of it. Okay. Okay, so now let's talk about the phospholipid. What is important about the phospholipid? They have the phosphate head and the lipid tail. And we know that our phospholipid bilayer of the cell membrane, right? Lipids are facing each other, and phosphates are facing the what? Phosphate is polar and facing the outside water molecule, right? Okay. What about the steroid? Steroid is also a very important example of the what? Lipid. So cholesterol, vitamin D, steroid hormone, bile salt, these are all the example of the steroid. And the last one, icosanoid. There are so many different one, like arachidonic acid, which is part of your cell membrane, and there are some which are prostaglandin, which can play a very important role in blood clotting, control of the blood pressure, inflammation, and the labor contraction. Okay. So now let's talk about some review question. What are the monomers of carbohydrate? Glucose, monosaccharide, glucose, fructose, and galactose. These are all monosaccharide. What is the name of the bond between the, okay, what are the monomers of protein? And what is the name of the bond between the amino acid in the protein? Peptide bond. Distinguish between the primary, secondary, tertiary, and the quaternary level of protein organization. When we say primary, what do we mean by that? Polypeptide chain, right? Polypeptide chain. When we say secondary, they will form the what? Spiral or sheet, we call it secondary structure. And what about the tertiary structure? When one polypeptide fold, we call it tertiary. When multiple polypeptide fold, we call it quaternary. What are the structural differences between RNA and DNA? RNA single-stranded, DNA double-stranded. Which sugar RNA has? Ribose sugar. And which sugar DNA has? Deoxyribose sugar. What are the bases RNA has? Adenine, guanine, cytosine, uracil. And what about the DNA? Adenine, guanine, cytosine, and thymine. Okay, so that's all about this chapter. Does anybody have any question?